It is all being recorded. Everyone, we're here. Woohoo! Blair's going woohoo in the chat room. I'm just waiting to make sure that we get the signal from the Bat Cave that everyone is hearing us and that we can move forward with our broadcast. Oh, there it is. There's that wonderful signal. And so, starting in 3, 2, this is TWIS. This Week in Science, episode number 687, recorded on Wednesday, September 12th, 2018. Nature's Tangled Tree. Hey everyone, I'm Dr. Kiki, and tonight on This Week in Science, we are going to fill your head with wolverines, a sugar maker, and tangled trees. But first...
Disclaimer. If you don't believe in evolution, you are wrong. Even if you haven't replaced evolution with anything specific or supernatural or vague. By choosing to go against all observations across all domains of science, you have chosen to be wrong. If you do believe in evolution, congratulations, though you are likely wrong as well. Not as wrong as someone who outright denies that life forms evolve, but your understanding of what evolution means, how it works, how it's taken place over the past 4 billion years is not likely a model that fits very well with how evolution has actually taken place on this planet. But fear not, you are not alone. In fact, you've probably got a better grasp than Lamarck, Darwin, and Huxley had. Not that they were wrong, you just happen to have been afforded an extra 100+ years of more scientific insight and discovery to base your initial impression on. And tonight, we will delve deeper into how evolution happens, where we've been in our search for answers, and maybe where we are going. All that and more ahead on This Week in Science, coming up next. Where's the music? This is an awkward pause between when we were supposed to have...
This
is what gets edited. This is where we edit out silence.
Music, cue the music.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin and Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again. The gang's all here, ready to talk about science. So thrilled. This is one of my, favorite evenings of the week. The highlight of my week every week, I most happily can say. What do we have in store for you tonight? Well, I have sugar. We got some sugar for you. Got new neurons. That's always good. And we have an interview with author David Quammen. Justin, what did you bring?
Oh my gosh. I have got beer before there was farming. I've got elephant birds and ancient humans. I've got the first human drawing and human blood with mutant powers.
What? All right, let's go with that. And in the animal corner, Blaze.
Blaze.
It's Blair. And I have a case of invertebrate kidnapping. I have confusing chemical mimicry. And I have sharks in Blair's animal corner.
Okay. All right, everyone. Looks like we have sharks. No, we've got a great show lined up for you tonight. And before we move on, though, I would like to remind you all that if you have not yet subscribed to the This Week in Science podcast, it's easy to do so. We are all the places podcasts can be found. Look for This Week in Science, or you can easily find us at twis.org. That's twis.org. That's pretty easy. But now it is time for our wonderful guest interview for the evening. Our guest tonight is David Quammen. He has studied at both Yale and Oxford, which means he's really smart. And he has spent his life writing amazing, and I will say amazing, stories about nature, evolution, and man's relationship to the natural world. He contributes to National Geographic magazine and can claim to have been the sole author of one issue, which is a claim you can only aspire to in your dreams. He wrote The Song of the Dodo, Monster of God, The Reluctant Mr. Darwin, Spillover, Ebola, The Chimp in the River, and a book about Yellowstone, and most recently, his book, The Tangled Tree, which we are thrilled and honored to speak to him about tonight. David, welcome to the show. Thank you for joining us.
Dr. Kiki, great to be with you. Great to be with you, Twissers. What have I got? I've got horizontal gene transfer. I've got endosymbiosis. I've got tangled trees. I've got transposons. I've got Lynn Margulis, Carl Woese.
Oh my gosh.
Whoa.
You're teasing up a storm here. Yes, you
Let's go.
Let's go. Yes, all these things now. All right. You have written about the natural world for years, and
Decades, yeah.
Decades. And I first did become aware of your writing through The Song of the Dodo. What is it that draws you to write about the natural world? Like, what inspires you to keep writing amazing book after amazing book?
Well, I am deeply interested in evolution, ecology, conservation, biological diversity on planet Earth, following brilliant scientists, men and women, through jungles and swamps, getting my feet dirty, seeing Komodo dragons running up cliff sides, and learning about the mysteries of molecular biology as well as every other kind of biology. I mean, I started as a fiction writer, and then I gravitated into nonfiction, and the natural sciences became a journalistic beat. So I've been doing this now since about 1980. And I don't have a job, but if I had a job, this would be the world's best job.
It would be, could be,
Yeah,
might
would be, yeah. But I don't have one. I'm a freelancer.
But you have so many probably amazing stories from your experiences. I was reading about one of your journeys hiking a transect.
Oh, this was the greatest privilege of my life. I was asked back in 1999 by National Geographic to do a series of articles about a guy, an American ecologist named Mike Fay, who was going to walk 2,000 miles across the Congo Basin doing a biological survey on a zigzag path, off-trail bushwhacking to identify what was there, what were the biological hotspots, and what were the places that were less crucial. So I spent weeks walking with him. We walked in sandals and river shorts through swamps and jungles and swam across black lakes, and I wrote 3 stories for National Geographic, and I had a wonderful photographic partner, the great Nick Nichols. Mike Fay was the explorer, Nick Nichols was the photographer, and I was the writer. Great memories of that. The Mega Transect, it was called.
So the first thing that occurs to me is there's a guy who's willing and able. He knows that he can survive on a trip like this, and he's going to make this voyage. And then they assign 2 other people who may have no experience bushwhacking or surviving out in these kinds of conditions for was that a thing? Like, did you have a lot to learn then, or were you already prepared for a mission like this?
Well, it was not a random choice that they picked Nick Nichols as the photographer and me as the writer. They picked us because they hoped the right people for this sort of a challenge. We weren't as tough or as crazy as Mike Fay, but we were a close enough approximation that they hoped we could hang with him at least for long sections of this walk. He alone did the entire walk, 456 days on the trail in the midst of Central African forests, without a hot shower, without sleeping under a roof, and doing great work censusing what was there. And I walked for about 8 weeks with him, divided into about 4 stretches. So, 53 days, I think it was, which is nothing compared to him. But 53 days of bushwhacking through the Congo will
That's
we'll
substantial.
We'll give you a sense of the experience. That'll give you the flavor of it.
I feel like I deserve a beer after an afternoon hike. So
The greatest part of the greatest thing about this was that I learned to protect my feet by duct taping my feet every morning before I put my sandals on and left the tent. I duct taped over all of my cuts and sores. I iodined everything, and then I duct taped over them. And I learned that was the way to survive walking in the Congo. And then when Jane Goodall came to join us, and she walked for an entire day to get into one of these sites, and her feet were totally torn up from the cheap plastic sandals that she was walking in. She turned to me. She calls me David Q. She knows a number of Davids. She's got a very complicated, busy life. Yeah. She calls me David Q. And she says, David Q, would you duct tape my feet?
So first of all, why aren't you wearing shoes?
Because shoes fill with water and mud. And because long pants can never be kept clean. If you're bushwhacking across the Congo, through swamps, across streams, etc., through mud, it's very difficult to keep socks dry. It's very difficult to keep boots dry or have them high enough, because sometimes you're in up to your waist or your armpits. The easiest thing to keep clean and dry is skin.
Yeah. Wow.
So you go ahead and let your skin breathe, and then you just repair your skin as necessary.
Up to your armpits? I mean, isn't there there's just points of no walking around something?
No, yeah, right. We were following Mike Fay's compass line.
He was going
to do the transect.
And sometimes you walk across a stream, and it's a blackwater stream, and you go in up to your waist, you go in up to your armpits. You come out the other side, you walk through mud, you come out with mud all over your feet, and then Gesundheit blaze. And then you spray water over your feet to rinse them off. And sometimes you come to a lake, and the lake is too deep, so you swim across the lake, and you fill your pack with your waterproof pack. You fill it with air and close it tightly, and you push it in front of you like a beach ball, and you dog paddle through the mangroves, and you get to the other side of this lake. We had a great time. It was the greatest experience of my life.
Yeah. But the bushwhacking across Africa through the Congo, I mean, this is a far cry from molecular genetics.
Yes. And that's what made this book so difficult.
All right. So how did you come to this book and explain some of your difficulties there?
Well, so most of my books have been about ecology, evolutionary biology, conservation, and they've involved a lot of outdoor adventure, like I've just described. My book, The Song of the Dodo, it's about islands and evolution on islands. So you're walking around on Komodo, and a Komodo dragon jumps out of the bushes and runs up a cliffside, and that you have some storytelling. This book is about, I hope this is a page turner, and it's about a radical new history of life on Earth. But another way of saying it is that it's a history of molecular phylogenetics. Oh my God, a history of molecular phylogenetics. That sounds very dry. The only jungles I'm walking through are jungles of genomic data. And that made this book probably the riskiest and most ambitious that I've ever written. Not physically, but intellectually. And it all began in 2013 when I first read the phrase horizontal gene transfer. And I said, no, that doesn't happen.
What?
What? Wait, what?
Genes do what?
Yeah. Genes move sideways across species boundaries from one kingdom to another? No, I've written enough books about evolution that I've got 5 reasons why that can't happen.
Yeah.
And then I read into it further, and I found, yes, we now know from genomic data, from gene sequencing and bioinformatics, high-powered computer analysis and comparison of genomes, that this is a thing. This happens. Horizontal gene transfer, genes moving sideways from one great limb of the tree of life to another.
Yeah.
And that's where it started.
We've talked before on this show about the horizontal gene transfer that occurs between bacteria.
Yes.
So bacteria can get antibiotic resistance because they get those genes from other bacteria that happen to have it.
instantly.
Yeah, which is amazing. And then the idea that, plants also do this, but then, like you said, between kingdoms. So you've got bacteria to plants, fungi to plants. And then let's talk about between into the animal kingdom.
Viruses dragging DNA from other creatures into animals. Viruses depositing their own DNA in genomes, including the human genome. We could talk about the fact that we now know that 8% of the human genome, roughly, is, excuse me,
Yes.
no more boings because I just closed my email program. 8% of the human genome is viral DNA from captured retroviruses that have come in sideways, not recently, most of them, but over the last maybe 100 million years, but not down through the long linear sequence of animal evolution, but sideways, interrupting that lineage, coming in retroviruses that have infected reproductive cells and therefore become heritable, composing now 8% of the human genome, including some retrovirus genes that now perform important functions in mammals and pointedly in humans, such as producing the membrane between the placenta and the fetus that makes human pregnancy possible, or successful human pregnancy possible. So it's a retroviral gene that has been repurposed, acquired sideways.
So we might still be laying eggs
Yes,
if it weren't for a virus, right?
Yes, exact. Might is important because this is speculative, but the wonderful scientist, French scientist named Thierry Heidemann, who with his group working at an institute on the south side of Paris, have explored one of these genes, a gene called syncytin 2, Syncytin, Cytin, syncytin 2, not as in Cincinnati and the Cincinnati Zoo, but with Y's. Syncytin 2 is this gene. It comes from a retrovirus, but now it's in the human genome, creates this membrane between the placenta and the fetus, helps to carry nutrients into the fetus from the placenta, helps to carry waste products out to the placenta from the fetus so the mother can get rid of them, and probably also protects the fetus from the mother's immune system, because of course the fetus is only 50% the mother's genome and the other 50% is this guy. And therefore the mother's immune system would attack it probably. Right. This magical membrane helps to protect it. And that membrane is created by a viral gene that formerly created the envelope membrane in retroviruses. It was acquired, it had been repurposed, and now it has an essential function in humans. And there are earlier versions of this, different versions of this, in other mammals. So it seems to be that there have been multiple acquisitions, multiple infective events that brought this kind of envelope gene into the human genome and gradually allowed animals to figure out a way to nurture their offspring internally rather than laying eggs on the ground. So this might be responsible or partly responsible for the evolution of mammals.
Yeah. Without that, pretty much all mammals would be different. It's completely changed their adaptations for survival because they can move with that thing that they're carrying inside of them instead of having to go back to a nest over and over.
Yes. A female can walk away from a predator, taking her young with her, rather than leaving them there on the ground and hoping, like a killdeer bird, that, if I go over here and make a bunch of noise, I can distract the predator. No, she can just say, we're out of here. Offspring and me, we're out of here. So that's what the evolution of mammals made possible. Probably a fairly important moment in the history of the tree of life.
Yeah. So
very likely arose at first as a defect of some sort. Like I'm trying to imagine this transition of now the egg isn't the shell maybe isn't forming, and then things are different. Like if we were diagnosing a human that was for whatever reason now forming a hard-shelled egg inside them. We see this is a disease. This is something we need to cope with and fix. But allowing evolution to take its place turned something that was egg-laying into a mammal.
Yes, but nobody knows, Justin, what those stages were. We don't have the data to resolve those questions, those very legitimate questions that you just raised. At least so far, we don't have the data. What we have are genomes, fully sequenced genomes that we can compare to one another. And that's what my book is about. The Tangled Tree is about the discovery that discerning the history of life, the history of evolution, could be done by sequencing genomes and comparing them one to another. Francis Crick, back in 1958, sort of launched this idea and called it protein taxonomy. His thought was not that you use genomes, but you might use proteins, another form of long molecule made up of these individual units, and you could sequence the units, the amino acids, and then you could compare one protein, like a hemoglobin protein in a horse, to a hemoglobin protein in a human, and you could see how long ago maybe it was that humans and horses had diverged from each other. That's sort of the initial thing. But then this whole story that I tell, the idea of doing it with DNA and RNA.
Yeah, looking at it from the historical perspective, I mean, we talk a lot about, how once upon a time, and still to this day, people insist on morphology for categorization.
Yes.
And you go through the long history of the development of the tree of life, and how Darwin changed the meaning of the tree of life and what that came to be, and then molecular genetics.
Right. Yeah.
Changing it yet again.
Yeah. And molecular phylogenetics, using long molecules to discern phylogenetics, discerning the shape of the tree of life, who's related to whom, and how long ago did branches diverge. Yes.
Yeah. So to you, as you were going through your process of, researching this book, writing this story, and basically telling this history, like, what really stood out to you as some of the, or one of the most pivotal moments in the history?
Well, a couple of pivotal moments. I mean, I started from the end. I started because I picked up the thread of this phenomenon, horizontal gene transfer, in 2013. I'd never heard of it before that. Maybe I read a blog by Ed Yong. Have you ever had it? You probably know Ed Yong.
Yep.
Yeah. So it might have been one of Ed Yong's blogs, and he mentioned this. And then I said, I got to read into that further. And then I discovered a scientist named Ford Doolittle who had written about this in review papers. And then I discovered this scientist, Carl Woese, who launched this revolution in doing molecular phylogenetics, using molecular data to discern the tree of life. And I went back to Lynn Margulis, whom I knew. Lynn Margulis, the great microbiologist of the second half of the 20th century. And she had been a morphologist also, but a micromorphologist. And she was making important discoveries and rediscovering ideas, like the idea of endosymbiosis, that complex cells have become complex by swallowing bacteria and turning those bacteria into internal organelles. She revived that theory and propounded it beginning in '67 for a long time, and people thought she was wacky, thought she was crazy. And then others came along with molecular data, molecular methods, and said, well, let's check this using let's sequence the genome of a mitochondria and see whether it looks like a bacterium or not. So people like Ford Doolittle did that and said, she's right. She's absolutely she's right. And she's right about chloroplasts in plants too. They are captured bacteria. Endosymbiosis, lineages have come together. Branches on the tree of life have not just diverged, but converged. And that began this whole rethinking. And then Carl Woese comes along and he invents a new method of doing this, and people follow his method. And then they discover horizontal gene transfer is going on all the time among bacteria, and it's happening also amid animals, and the tree of life becomes very, very tangled. So crucial moments. Lynn Margulis, 1967, re-announces the idea of endosymbiosis in this paper that gets rejected by 15 journals, according to what Lynn says, and eventually gets published. In 1970, she publishes a book. She's out there. She's wonderful. My God, Lynn Margulis. And then 1977, Carl Woese hits the front page of the New York Times above the fold with his discovery using his primitive genome sequencing method that there is, in fact, a kingdom of life that nobody knew about before, a completely unknown, obscured kingdom of life. Everybody thought there were two great limbs on the tree of life, bacteria and everything else, the two great kingdoms of life. Bacteria, which were simple cells, no cell nuclei, no internal organs, no internal complexity, and everything else, the eukaryotes, the true kernel, eukaryote coming from the Greek true kernel, because they have cell nuclei and they have internal organs like mitochondria. And
it came from the bacteria that were the other branch of
back
life.
to Lynn. Yeah.
There.
So 77, Woese hits the front page of the New York Times. You know, Andy Warhol said that in the future, everybody will be famous for 15 minutes. Woese gets his 15 minutes on the morning of November 3rd, 1977, when the New York Times has a picture of him sitting at his desk at the University of Illinois in Urbana in a sports shirt with his feet in Adidas sneakers up on his desk, and they say scientists discover a separate form of life resembling early life, and that's this third form of life which became known as the archaea. Archaea as in archaic, as in archaeology, old, because we thought these creatures might reflect the lineage of the oldest creatures on Earth. They look like bacteria. Through a microscope, Lynn Margulis or any other microbiologist took them for bacteria. But if you sequenced their genome, suddenly you saw, bingo, these things are not bacteria. They're very distinct from bacteria. And in fact, they're more closely related to us, humans, animals, plants, fungi, than they are to bacteria. So he declared that they were a new form of life, a third form of life. And that was the beginning of this whole radical redrawing of the tree of life.
Yeah, and oh, go ahead.
Not rejecting Darwin, not refuting Darwin, not falsifying Darwin, but adding some very important things to the Darwinian understanding of evolution that Darwin and the neo-Darwinists of the 20th century, the first 2/3 of the 20th century, had no chance to recognize.
Yeah. And now we're sitting here looking at all of this molecular phylogenomic data and saying, well, Darwin was right. We know natural selection does work, but we also know that, in the human genome, for example, we have multiple genes responsible for a particular trait. So when a gene mutates, maybe it's silent for a really long time. And so there are all these complex factors that work internally in terms of how rapid mutation occurs, how it changes. And then you've got the things like this infectious heredity.
Infective heredity, Joshua Lederberg's term for this. Yeah, because he was studying this among bacteria in the 1950s, and he and Esther Lederberg and Norton Zinder called it infective heredity. And now we know it occurs not just in bacteria, but in all forms of life. But yes, and it tells us, it reveals that yes, natural selection is still the crucial thing, the great Darwinian mechanism for how species evolve and adapt. But what's different, radically different, is the sources of the variation among populations upon which natural selection acts. You mentioned, Kiki, there's this incremental mutation. DNA replicates as species reproduce themselves, and DNA makes these little tiny mistakes. It gets a letter wrong here and a letter wrong there, and there are these incremental mutations that create variation among the individuals of a population. And then there's competition, and there's the survival of the fittest, and natural selection allows the fittest random variants to survive. all still true. But what's drastically new is that the sources of variation we now know include not just these micro mutations, these little incremental changes as DNA copies itself, but whole packages of DNA that come slamming in sideways by horizontal gene transfer and present major new possibilities for natural selection to work on, as with, as you've said, bacteria acquiring antibiotic resistance. They can acquire an entire gene for antibiotic resistance. One strain, E. coli can acquire a whole gene for resistance to erythromycin that may have evolved in Staphylococcus and then been transferred sideways. And that happens in
all of this.
In animals too.
Yeah, and all of this. I mean, we question
and we're
on
proud
this
of,
show.
of text messaging and email in the modern age.
Right?
that's nothing to the type of communication that is taking place in
This
the bacterial
genetic
level.
communication, right?
Yeah.
And we argue all the time on this show at the macro level about what a species is, right? We have subspecies and there's hybridization, all of a sudden a species, because of an ecological event, suddenly these two diverse populations are brought together and lo and behold, they can breed. And we thought they were separate species, but they're not. And so biologically, behaviorally, we have this, I guess, diffuseness to the definition of a species.
Yes.
But what you're talking about adds even more.
This is, yes, totally agree. This is very important. I mean, anybody who's read a little bit about the way organisms are classified maybe knows that there are species, genera, family, orders, classes, kingdoms of life. And those are nested categories of how we organize our knowledge of life on Earth. It's not the way life on Earth organizes itself. It's the way we organize our knowledge. And we know that those larger categories, kingdom, phylum, order, family, class, family, et cetera, those are artificial human groupings for our convenience. But when we look at it, but we tend to believe that species is an actuality. It's a real category. It exists in nature. But the closer you look at it, the more you have to admit and you realize that even the category of species has blurry lines. I say near the end of this book that there are three categorical ideas that people have embraced. The idea of species as discrete entities. They are populations of individuals, but they have fixed memberships and they're separate from other such entities. The individual is a discrete entity. There's an individual, Dr. Kiki Sanford, Charles Robert Darwin, a brown dog named Rufus, individuals. And the third categorical is that the history of life is shaped like a tree, coming from a single origin, branching, diverging over time, growing up, creating diversity. And now we know that those three categoricals, species, individual, and tree, are all wrong.
They're all
there you go.
So
basically, but don't mistake. They're not totally wrong.
Right.
They're just not categorically right. They are not categorically right, and in that sense, they are wrong.
It's gonna be like that. I think there's a scene from Princess Bride. It was like, "Is he dead? Well, he's mostly dead." These ideas—they're mostly dead.
Exactly. Yeah.
So life is less of a tree and more of like a tangled ball of spaghetti or something like that.
Exactly.
It's something else. It's something else. Yes.
The term I've come to, and I don't know who's coined it, is the braided stream, and it's mostly applied, I guess, to human evolution or hominin evolution, because all of those things that, again, we learned through morphology and the idea that there were distinct handoffs in this relay race of becoming a modern human from a species of human to another species of human, where we find that there's lots of interbreeding going on throughout the ancient hominin history. And it's getting harder and harder to point to any one period of human evolution and say that's its own distinct, completely removed from the rest of the hominins that were running around.
Because now we know that Neanderthals bred back in with humans, Denisovans probably bred back in with modern humans.
And Neanderthals.
Yeah,
that was the recent
Neanderthals,
story.
yeah. And even chimpanzees bred back in with the human lineage after the divergence. We know that from molecular phylogenetics. Yeah.
Absolutely.
But this is even more radical. This is bacteria breeding back in with the animal lineage. This is viruses breeding back in with the human lineage.
And so then the question is, where do the viruses come up with this DNA? Right. So
well, nobody knows where viruses come from. Viruses are still a wonderful mystery.
They are.
There are ideas. There are thoughts.
There was a story a few weeks or months ago that we were talking about where some of the giant viruses were being seen to sort of create new series of genes that weren't present in previous versions or in related versions of these viruses, so that they were actually doing a little bit of unique gene creation with them, as though they were there unto themselves, their own genetic laboratory, sort of creating places.
Viruses are the breeding ground of all genetic diversity.
So no, but I mean that's one.
Okay, ladies and gentlemen, you heard it here from Dr. Kiki.
That's right.
I didn't say it; she did.
I'm wondering, though. I mean, this book, I mean, you're bringing to the public eye and to the public mind these radical, as it says, the radical new history of life, these radical new ideas that are, really becoming the fabric of our modern evolutionary thought and how we consider things. I mean, do you think that there's that this is the beginning of maybe a philosophical questioning? If more people start thinking about this, that it might raise.
I hope so. I mean, I've been talking to audiences, live audiences, for the last month or so, and I describe this stuff, and people say what I said in 2013. Wait, what? So I hope a rethinking, at least of those three categoricals that I mentioned: species, individual, and the history of life. I mean, and sort of the bottom line of this is a reaffirmation of that great dark truth of Charles Darwin, which I think of as the darkest of his truths, which is that we humans are not separate from nature. We're not this holy thing that sits apart from nature, somewhere up in the sky above nature. We're part of nature. And Darwin's work dramatically announced that. But this stuff announces it even more challengingly and dizzyingly, but it reaffirms that, and it brings us back in to, I think, a position of humility and engagement with the rest of the natural world that is salubrious and crucial as we deal with our attempts to maintain a viable environment and situation for all kinds of creatures on this planet.
Yeah, all the creatures, big and small, and ourselves among them.
Yeah.
So what do you consider yourself? Individual? How do you look at yourself now that you have this knowledge and this new perspective on the world?
Well, 8% viral, maybe 3 or 4% bacterial, 80% animal, husband, Montanan, science writer, etc. But and plus the microbiome, plus all those other critters that are living in us that are not part of our genome. You've probably talked at great length about the microbiome on this show with Ed Yong and maybe Carl Zimmer and other people. All those other critters living within us. So we're these walking ecosystems, and we get to have a single name, and we get to live for 70 or 80 or 90 years in this amazing place, this amazing planet. And it's I'm not going to say it's a miracle, because I don't believe in miracles, but it's the evolutionary equivalent. It's this extraordinarily gratifying privilege to be part of this and to have what appears to be but isn't an individual identity for a while and walk around and say, hi, I'm Kiki, I'm Justin, I'm Blair, I'm David, I'm Blaze, and it's nice to be on this planet, even while knowing that you are a chimera, a composite of all these other things.
A mosaic, a chimera. I am the matrix.
Yeah. Yes. But that said, I also want to just mention that this book of mine, it's a history of molecular phylogenetics, but also it's a bunch of storytelling. I hope it's a page turner thing. It's about this wonderful character named Carl Woese, this wonderful character named Lynn Margulis, these other wonderful characters, Barbara McClintock, Ford Doolittle, et cetera, et cetera. It's about mysteries solved and lives lived and challenges met.
Yes, I think it is a page turner. I have been reading it myself and quite enjoying it. Something that's different. They're very short chapters, which I was like, wait, this is different. You've got these little tidbits, and you go instead of having these massive tomes.
I think about the reader. I think, what's the reader's attention span? What does the reader want? Does the reader want an explanation or a joke right now?
I want the story, and you do a wonderful job of bringing the scientists into the story and exposing their humanity and how they are going about solving the mystery. And so thank you for writing this book, and thank you for joining us on the show tonight.
Thank you for letting me talk with you tonight. This is great fun, and I really appreciate it.
You're welcome. We are enjoying it. Yeah.
I really do love the telling of the journey of the discovery. It's one of those things that I think the general public might just assume that scientific discovery is a natural thing that just happens as more time goes by. But it does take a lot of hard work and a lot of courage by people to go out and pioneer things, to make these observations, and to gut check themselves about what they actually have observed, and push forward with that, and to commit themselves to maybe even being somewhat ostracized by their peers for doing so. And it does take a level of courage and bravery, and a lot of hard work that I think gets overlooked when we only are touching on the highlights of what has happened and the highlights of what we have learned.
It's a good reminder that throughout history, people that were really super right were often called wrong first.
For a long time, sometimes.
Yeah. They laughed at Columbus. They laughed at Kiki Friedman. They laughed at Lynn Margulis. And they laughed at Soupy Sales.
He was a comedian.
And they were all right. They were all right. They were all
visionaries. They were all right.
All right. Where can people find you online if they are interested in learning more about you?
Easy. www.davidquammen.com, my website. I'm on Twitter @David Quammen. There aren't that many science writers named David Quammen. I'm easy to find.
People will be able to find you. Your book, The Tangled Tree, A Radical New History of Life. Once again, David, thank you so much for joining us tonight.
You are very welcome, Dr. Kiki and company.
And I'm sorry I missed you while you were here in Portland for that whiskey. Maybe I will catch up with you at NASW in Washington, D.C.
I had etouffee at Jake's.
Etouffee?
Yeah, at Jake's, yeah. Jake's great crawdad place.
Mm-hmm.
Anyway, thank you so very much. Thank you, Blair. Thank you, Justin.
Yeah, pleasure having you on.
Have a lovely evening. Everyone out there, we are going to take a quick break. This is This Week in Science, and if you would like to stay tuned for science news, that's what's coming up next. I've got sugar. There's some Wolverines, and Blair has a human shield. No, you've got parasites. Blair has parasites.
I have a kidnapped shield that is an invertebrate.
Okay, we'll trust you.
It's a lot to unpack.
It's a lot to unpack. We'll work on that. Yeah. Everyone, we'll be back in just a few moments. This is This Week in Science. Stay tuned for more.
and explain things you've heard with more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within this scatter plot.
Hey everyone, thank you for joining us for another episode of This Week in Science. We do appreciate your time and that you're spending it with us to enjoy the world of science with us. Now, we can't do this show without you. I mean, without you as an audience member, but also without you as a supporter. We need your help to maintain this show, as currently we are listener supported. We do not take advertisers at this point in time so that we can give you the best show, and we just want to do this for you. So, if you want to help support This Week in Science, how can you do that? Well, let me count the ways. 1, you can head over to TWIS.org. TWIS.org is our main portal for all things TWISy and sciencey goodnessy. Big thing is if you know somebody, meet somebody, and you're like, oh, there's this podcast I really like, This Week in Science, but and you're talking, and then where do you find that? Tell them to go to TWIS.org and help them find the big orange subscribe button. And if you're watching right now and you haven't subscribed, go to TWIS.org, click that big orange subscribe button. If you're on YouTube, click that subscribe button. But if you go to TWIS.org, you click on it, it gives you Google Play, iTunes, and YouTube, the big 3, so that you can find your preference for subscriptioning. So you get us every single week. That's right. Wouldn't you like to hear TWIS every week? I know I like doing it every week. Beyond subscribing, what else can you do? Well, you can help support us by buying our merchandise. There we go. Click on the Zazzle link at TWIS.org. It'll take you to our Zazzle store, zazzle.com/thisweekinscience. Head on over there. We've got mouse pads and t-shirts and pillows and hats and mugs and all tote bags, all sorts of fun things, so that you can enjoy TWIS in multiple forms and also help spread the word of TWIS. People might see it and go, oh, what's that? And check it out. So it might start conversations. Who knows? The thing here is that a portion of the proceeds do go to support us. So you not only get a cool TWIS thing from buying through Zazzle, like you need a new shirt, get a TWIS shirt through the Zazzle store. You get that cool thing, but you're also supporting us with a portion of the money that goes to buy that item. Back at TWIS.org, other ways, if you're not into stuff, and I totally get that, I'm trying to clean out my garage as it is, you can help us by directly donating. And you do that easily. There's a yellow button on the main TWIS.org page. It says donate. Click on that. It'll take you to a PayPal donation interface. Not a big fan of PayPal? Well, you can click on this link up at the top of the page that says Patreon. Click on that Patreon link, and it takes you to our Patreon community page where you can become a patron. So if you haven't done so yet, click on a donate button on our website, click on that patron link, and figure out a level of support. You can donate once with that yellow button, or you can donate in a recurring fashion through Patreon, where you are charged once a month at your chosen level of support. And in return, you get cool things from us that are like little thank you gifts. One of the big ones, and I'm going to say it, the $10 a month tier is where we thank you at the end of the show. We will read your name among the other supporters of TWIS on Patreon, and we will thank you by name. So if you want that $10 a month, it's basically like a cup of milky coffee every week. It's the same amount. I mean, do you like us more than a cup of milky coffee? Then support us at that $10 level every month. It's super easy. Anyway, TWIS.org is the main place where all of this, you can find all these things and help us out. Keep us going. Help support us. We can't do this without you. We thank you so much for your support.
ocean. A line freeze shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
And we're back with more This Week in Science.
Yes, we are. Hey, you know what time it is.
Ooh, is it
What
time
is it
for our
now?
favorite part of the show?
Yeah, time for This Weekend. What has science done for me lately? All right, this week is a very short note from April Robeson. April writes in to say, science has allowed me to be super extra and give my rescue puppy a doggy DNA test.
Oh.
I got to find out what he is made of, which isn't necessary, but is quite fun.
That's true. Science is fun.
Yeah. I
Science
did that
is-
once. I did that once.
To your dog?
Yes. Yes.
Oh.
This was a long time ago. But it was you could see the little bit of collie was there, maybe husky, and then it also turned out through the genetics that she was part of this. Now I can't remember because it was a long time ago. Part this Korean breed of dog that was very specifically this Korean breed, which was known for its intelligence and still had kind of some of the similar markings of a collie too. And the collie was in there as well, but it was pretty fun.
Is it the Jindo?
Yeah, I think that was it. Yeah.
I have never heard of that dog before.
And
wow.
this was
Okay.
the most intelligent dog I've ever known, and that's one of the things Jindus are known for. Is being
It's known for its fierce loyalty and brave nature.
What, did you just look that up on the internet?
Sure did.
It sounded like Quotey voice. Yeah.
It did. No, you're looking things up. April, thank you so much for writing in. I love the fact that we have these modern day single nucleotide polymorphism testing services. They're not checking all the DNA, but they're checking these little bits and pieces and going, how does that match up with this? How does it match up with that? And does it match? And does it match? And it's fun. It's fun to find these things out. And I'm especially when it comes to your own animals, being able to find out, especially dogs and cats, we don't know. So often they're mutts and mixes, and even when you think they're purebred, are they?
You got to be careful though, because once you have your dog's DNA tested, they can be easier implicated for a crime. So
That's right. They could be.
you gotta watch out.
Their cousins get tested, and suddenly the DNA traces back. Yep, right there.
Before you know it, Fido's implicated in that robbery.
Oh, Fido!
trouble.
Why? Why did you have to rob that
I
bank?
give you everything you needed. I did wonder where you got the sports car. That was odd.
Yeah,
That was
Came
odd.
up with a scratch for that.
Yeah, I wasn't expecting that, Fido. That was weird. All right, April, thank you so much for giving us this fun little tangent to think about. And yes, I do agree. Science can make things like learning more about your dog so much fun. Everyone out there, if you have a little note, if you have a longer note, send them to me. Tell me what science has done for you lately, and we will read it in this portion of the show. Share it with everybody, because really, if science is helping you and making your life awesome, what has it done for you? Is it making it? Let's share. Let's celebrate this influence that science has. You can go to the Facebook page for This Week in Science, which is This Week in Science, and leave us a message, or you can send me an email. Kirsten, KIRSTEN at thisweekinscience.com. Let's keep this portion of the show going.
Can I make a special request? I made a request for a sonnet, and we
Yes.
got one.
Yes, we
Made
did.
a request for a haiku, and we got one. So I'm doubling down. I would like a What Has Science Done For Me Lately? Very short song. Email to Dr.
Oh,
Geek.
Wow.
what has science done for me lately?
Yeah,
What
we'll play it on the
has
show.
Science done for me lately.
Send them in.
Yeah.
I know
Send
we
'em
have
in.
lots of talented listeners.
That would be
Send 'em
fun.
in.
Oh, I love that. Great idea, Blair. It's a challenge.
Thank you.
Challenge. Who will accept the challenge?
Yeah.
All right. The challenge right now is to talk about science news.
Oh yeah, that.
Yeah, so we were talking about life and evolution in the interview before the break. And one of the big questions about life is, where did it come from? You know, my own son is asking me this, where did life come from, mommy? How did we get here? And I'm like, I don't know. We don't know. And, I try and explain to him the whole RNA hypothesis. And he's 7. So he's like, okay. He gets a lot, though. But one of the big questions is the basic building blocks. Many of the basic building blocks of the materials that created everything are sugars. And where did these sugars come from? And we've been looking all over the universe. We found big clouds of formaldehyde floating in outer space. We have found clouds of more complex sugars, glycoaldehydes, glyceraldehydes, floating in space. So here on Earth, we're like, all right, how do these sugars come to be, and how do they remain persistent, and how does this happen? And so a long time ago, there was a Russian chemist back in 1861, and he figured out this chemical reaction called the Formose reaction. Formose is a sugar, formaldehyde to a sugar. And the formaldehyde reaction occurs when formaldehyde is added to water and a base. It's this Formose reaction, and it makes sugars. The problem is, if you leave it alone, then it turns into like a tar. Everything continues to react and break down, and it just is gross and not life sustaining or supporting. If everything keeps reacting and breaking down, it can't be a building block. And so while the Formose reaction has been this tantalizing idea, it doesn't explain clouds of sugars in space. It doesn't explain the existence of life here on the planet. And so researchers are still searching for that chemical reaction that could explain the existence of these sugars. Enter Peter Schreiner and his team at Justus Liebig University in Germany. And they have found a molecule, hydroxymethylene, which is a really, really reactive molecule that they showed to exist about a decade ago. And they have now shown that without any solvents, without water, and at temperatures near absolute zero, which would be the temperature in outer space, if it comes near formaldehyde, it will join with formaldehyde to create glycoaldehydes and glyceraldehydes, sugars. And these precursors can form ribose and other carbohydrates without turning into that black tar that was the problem. It is sustainable. It is hydroxymethane is reactive enough, but then the products don't break down in that negative way. So the question is now, is this the molecule? Is this the precursor? Can this explain all the sugars? Yes. The researchers say, we believe that the reaction not requiring aqueous conditions is superior to anything that has been suggested in the past. I call this carbohydrate formation in the absence of biosynthesis.
I love your quotey voice.
This is like my very, very stern quotey voice.
Yeah.
My I am right quotey voice.
Mm-hmm.
Yeah.
I've been saying it on this show for probably years that I think in our lifetime we'll have a real, pretty solid explanation for how life began. And I cannot wait for that to be in a textbook, ready to go,
Right.
handed to school children.
Look,
I think it's happening.
This kind of a reaction, there's probably still more testing and confirming that needs to happen. We need to find this molecule in space. We need to see this happening in space, but not just on space. here on Earth also, this possibility. It could be how the building blocks get their start.
Yeah. I mean, I don't know, because I've never lived in the cold of deep space. But I also figure I mean, it also seems though that you could possibly have much more limited reactions in space, so that anything that we're trying to recreate is similar conditions as we can create here on Earth, could have a completely different outcome when things are spread out over a gaseous nebula of space too. So I don't know. I don't know that you would end up with tarry balls in space.
But the interesting thing, not requiring water, so that these building blocks could form in space with the absence of water. And then if the building blocks formed here on Earth where there is water, does that mean that the next steps in the chemical processes that lead to the formation of, little mud membrane bound RNA cells, DNA cells, is that what it takes? Yeah, that's exciting. I think it's interesting. And then my other interest story for today is new neurons in our ears.
Wait, I thought neurons belonged in the brain and just stayed there all the time.
These are interneurons. These are neurons that transmit information between the hair cells and other neurons. They are called spiral ganglion neurons, spiral ganglion cells. And originally, looking at the inner ear, the cochlea, and what cells connect to the hair cells, connect to the brain, all this stuff. Originally, we only thought that there were a couple, type 1 and type 2 cells that connected in there. But that was based on morphology, what they looked like. So going back again to this discussion from the interview, morphology versus molecular investigation. Researchers at the Karolinska Institute in Sweden have looked at RNA in the cells of the ear, and in doing so have discovered that there is not just one type 1 neuron, but 3.
Wait, 3 of just the type 1?
3 of just the type 1. And so they all serve, with their different RNA profiles, they all serve different functions. But what they seem to do is encode information about intensity. So the strength of a sound, the loudness of a sound, right? And the researchers now looking at this, using this single cell RNA sequencing technique that they have used, they can understand the auditory system better, maybe figure out drugs that can be used to treat things like tinnitus. It's amazing.
And I didn't bring it, so I won't go into it in any depth. But was just reading something that came out this week about a study where mice were able to tune out the sound of their own pitter patter, so that they could be more alert to. So it's also it's reading intensity, but there must be this feedback of identifying what that is and tuning down certain intensities so that you can ignore certain sounds, so that you can be more attuned to the sounds that are then important, and where does that prioritization take place. But also, this does, like you just said, open up a ton of pathways to treating ocular, not ocular, what's ear? What's ear? Auditory problems.
Auditory problems. Yeah. And
so you have your 3 neurons. You have your Dr. Kiki and your shouty Blair and your Justin. Your 3 kind of levels. You got to be able to mix all 3.
There you go.
Yeah, and maybe they work. Maybe they work in combination. Maybe they work separately. But these neurons, the way that they work, we used to think it was just one neuron doing all the work, but now it's these 3 different types potentially working in concert in synchrony to help us, focus on one person's voice in a loud cafe or at a party, be able to filter out the background noise to be able to focus on one particular thing. These cells are probably involved in that, and knowing that they have differences and that they're there and are different is going to make a huge impact in our understanding.
Yeah, and there's always my an initial reaction I sort of get to finding out that things are much more complicated than we thought before, which is like, oh great, there goes everything we think we know. But really what happens is when you sort of clarify and can sort of break down maybe a division of labor like you're suggesting between these, you can actually simplify your inquiries into things that you might be interested in. It might actually create much more clarity by discovering these complexities than was there when you didn't know that complexity existed.
Exactly. Do you have a story, Justin?
Oh my gosh, have I got stories? What do you want to hear? What do you want to hear? I got all these. Okay, here's what I'm going to start with. Just because, so I've got like 3 stories this week that sort of push back time points in history for things. We've been doing this over and over again. This is in Madagascar, strange and marvelous island off the eastern coast of Africa. A land untouched by humans until about 3,000 to 4,000 years ago, or so we thought. Now the Zoological Society of London is pushing back the date of humans' arrival in Madagascar back another 6,500 years.
But they're a zoo. What do they know about humans?
Right, exactly. What is the Zoological Society doing, coming up with discoveries in human history? Well, because this discovery is thanks to the missing toes of an elephant bird.
What?
Oh, snap.
So the elephant bird is some old school megafauna fowl. This at its time was the biggest bird on planet Earth. And we think of the biggest bird that you can think of, and you might think of
ostrich.
Yeah, kind of big. It's about as tall as a human. Probably weighs about as much, I would imagine. Now, the elephant bird was 10 feet tall, weighed over 1,000.
No.
Weighed over 1,000 pounds.
Had eggs.
Weighed over 1,000. You froze for a second. Weighed over 1,000 pounds.
Weighed over 1,000 pounds. Had eggs the size of a human head. Eggs, which if you cracked one over a campfire, would plop out about the volume of 12 dozen eggs onto your campfire griddle.
Which an ostrich egg is about 3 dozen eggs.
Yeah. So it's many more eggs.
Yes. Many more eggs.
So it's known that it went extinct about 1,000 years ago. And, it overlapped with kind of the humans, some of the more recent pinpointings of humans getting there. So it looked like, okay, human hunters got there and they killed all the elephant birds. That's what happened. But, now if you push back the history to 10,000 years, that's almost, it's 9,000-ish plus years of humans and elephant birds coexisting in Madagascar. Then what becomes maybe more prominent is the idea that they were stricken by diseases when they became exposed to domesticated fowl, like chickens, that sort of did arrive more in that 1,000-year ago time frame, and weren't native to the island and may have introduced things that could transfer, illnesses that could transfer to the larger fauna. But the answer may still be elusive. So this is, they made observations of ancient elephant bird bones. They revealed cut marks and depression fractures consistent with hunting and butchery by prehistoric humans. They used radiocarbon dating techniques. They were able to determine when the giant birds had been killed, and that's what pushed the date way, way back. Mm-hmm. So what's also interesting is these bones were found initially back in 2009 in a marshy fossil bone bed that had a large concentration of ancient animal remains. And it was thought this is just a condition where a lot of animals fell into a marsh and died.
Yeah, it's like quicksand.
Yeah. Now that they're seeing some of these bones that are being finally analyzed showing signs of butchery, they may need to revisit this entire site as a possible ancient human habitat site.
Yeah, that might be a trash pile.
Yeah. Or right, this might be a hunting ground, or where they bring everything back to chop it up and cook it, or
the compost.
Not even. Who knows? Huh.
Or maybe it was a combination. Maybe it was a trap of some kind, and also, you know, if it smelled enticing to carnivorous animals, you know, they'd go get stuck in there. But then it'd also be a nice little compost pile.
Right. So there's a bunch of possibilities. They need to go and reinvestigate. What's also interesting is that the peoples who are in Madagascar today aren't going to be related to the peoples who were there over 10,000 years ago. Right. These people we know migrated there 2,000 to 4,000 years ago. These ancienter humans, we don't know anything about, other than we're pretty sure they have no relation to the peoples who showed up later. So
this could be a little bit of a window into those peoples and their habits.
I'm not too surprised to hear that people might have come to Madagascar more than once, because one of the theories for how lemurs happened is that a prosimian took a raft. So
that sounds like okay. How did the lemurs happen? A prosimian took a raft.
Yeah. It's it's like why'd the chicken cross the road? It's one of the theories is that the predecessor to all primates took a raft to Madagascar and didn't evolve any further because there were no large predators. Because there's not really my understanding is there's not really a great fossil record for predecessors to lemurs on Madagascar, which makes them think that they came there, right? So if lemurs can hop a raft to Madagascar, certainly ancestors to humans could hop a raft to Madagascar.
Or maybe walk there. You're talking, if you're going back far enough, you're talking about enough ice age affects to make that a potentially walkable, or it's not a long trip.
How long ago was it? There was a period of time where the sea level was like 120 meters below where it is now. Like Yikes. It was very highly significant difference. But yeah, I don't remember when that was.
Yeah, then we were talking more the 20-ish thousand year ago. So I don't know when lemurs got to Madagascar. It says here that that's part of how they had been pegging human existence there. They know that there were giant lemurs and giant tortoises that became extinct within the last 1,000 years.
Quite a bit before that. That was about 50 million years ago.
Yeah. So
they think that lemurs, fossa, and most Madagascar mammals got there via natural rafts.
Yeah.
Wait. 50 million? Could it just been plate tectonics? Now you're talking a really long time scale. Where was Madagascar then?
50 million? No, there was still water. 50 million.
Where was Madagascar? I don't know. Yeah. Now I need to see one of those maps. Oh, science.
Oh, science and the changing plates. What else you got, Justin?
Okay. Here's another one. What came first? Beer or bread?
Good question. They both use yeast.
We found out that cheese came first last week. Right?
Yeah, we did bread. Bread got pushed back like 7,000 years.
Right?
I thought it would be beer,
years.
right? Because that's how people stayed hydrated.
Yeah, you have water that accidentally gets in with your wheat with the yeast in there. And look, what's this? How come I feel so
Okay.
funny?
Okay, I'll give you another one then. What came
Yeah.
first? Beer or farming?
Oh,
Beer.
So,
farming.
beer came first. And actually, we figured out bread came before farming too. Farming of wheat seems to have shown up as a result of being dedicated to beer and bread. And what's interesting about this story is this is taking place with the Natufian people, who are the ones that pushed back our understanding of the earliest bread. It's the same place, the same peoples. Turns out they had beer the same time they had bread. It's kind of
And
hard to
cheese.
tell which one of those came first now.
Beer cheese. Sorry.
It sounds like they had the
Continue.
makings. They had the makings for a wonderful diet.
Yeah, for a wonderful pub.
Yeah, their pub was doing
That's-
great.
yeah, that was the
Beer,
first-
cheese, bread, and beer.
that's
And
the
then they had a
first
pub,
thing.
and then people said maybe we should have a farm since we have a pub.
Yeah.
Yeah, it says beer findings from this Natufian site in East Jordan could be from 11,700 to 13,700 years ago. That beer, though, maybe not the beer that we're accustomed to.
You don't say so, huh?
Yeah, it was most likely a multi-grain thing where they just sort of threw a bunch in. It was probably more porridgey.
Yeah, I was gonna say fermented oatmeal.
Right.
Ew.
But
Yeah.
and my first sort of reaction, because they got a lot of this from residues, right? Recovered from stuff. My first reaction to this was, well, you found the oldest fermented something in the place where we just found out they had been making bread 11,000 to 15,000 years ago. So could it just be an accident?
That's what I was wondering. Also,
Yeah.
Bleak in the chat room says just rotted stuff.
Right.
Right.
So they actually dug quite a bit deeper, as scientists tend to do. So they had let's see. So the researchers believe that the Natufians used a 3-stage brewing process. First,
wow.
starch of wheat or barley would be turned into malt. That's just letting grains germinate in water. Then it would be drained, dried, stored. Then the malt would be mashed and heated. And then it would be left to ferment with the airborne wild yeast. They went through and tried to do a sort of reenactment. And in their reenactment, they did everything from pounding grains and grinding and doing all this stuff. And they found the traces left on the ancient stone mortars that they would find closely resembled their own lab experiments of pounding and crushing grain seeds. So it looks like, it looks like a lot of their recreations matched what they were finding in the field. What's also interesting is that they believe that this was sort of ritual beer use, because they would find the beer making components, the brewing equipment, near grave sites.
So ritual.
Yeah, so
Ritual
they would,
production.
you'd lose somebody, you'd
Yeah,
bury
but
them,
have a wake.
and then it would take a little bit, but you'd have a porridgey beer wake for them afterwards. And this is the researchers who were involved in this. Let me go assess their names. Professor Li Liu of Stanford and the doctoral student Jiang Wang have we probably talked about them on the show before. Back in 2015, they were looking at 5,000-year-old brews in China, and following brewing and the techniques that they've used and the discoveries that they've made. They turned their attention to this site and made yet another discovery, pushing back brewing by thousands of years.
I love it. Again, looking at the molecular aspects, looking at these little traces left over. But seriously, what do you do for your research? I study the archaeological evidence of beer.
Hmm.
Awesome. That's great.
And I've got one more pushing back story
Okay.
before we get to the
Push
corner.
it way back.
We'll get there, Blair. This is humans had been the earliest originators of art. For a long time, we created the first artwork the planet had ever seen. And then it turned out that the artistically impressive ancient human cave paintings in Spain were made 24,000 years before humans had arrived in Spain. And all credit went to Neanderthals, which meant that they beat us in artwork also by 20-something thousand years. But now we're back in the game. Scientists are pointing to the Blombos Cave in the southern Cape of South Africa, a drawing which consists of just 3 red lines crossed hatch with 6 separate lines, was intentionally drawn on a smooth rock flake about 73,000 years ago. That's 9,000 years-ish before the cave paintings. So we're first again. And how do we know these little markings are actually intentional and not just some sort of natural formation? Well, they used microscopes and electron microscopes, and they confirmed the lines were applied to the stone. Also, at the same site, there were other artifacts of symbolic tinkering, shell beads covered with ochre, engravings of abstract patterns on things that kind of resembled the drawing. They think the drawing was part of a larger piece of artwork, and that this is just the surviving fragment that they found. And they believe it was drawn on by an ochre crayon. And while this is amazing to truly reestablish the long human history with crayons, and I do hate to be an art critic ever, but it's nothing like the post-neo-biomorphic Stone Age representational perspective work of the Neanderthal cave artists. I'm sorry. Let's be clear. These humans were making zigzags with crayons, which you can call it drawing if you want. But I have a 5-year-old who can do much more realism with a crayon than they were doing in this cave, right? Much better skills than my 5-year-old than they had. And they were still...
Maybe they're just more into impressionism or abstracts, It can be very complex.
They were 30,000 years of art school away from even understanding the basics of what Neander artists of the day were putting out, cave after cave opening.
It's just like those white canvases you see at your local MoMA. You just don't understand it.
But maybe all we're seeing, this isn't necessarily, the important art of the time. Maybe these are just the doodles, the notepads.
I do a lot of patterns like that on my pads during boring meetings.
This is the equivalent of the marks left by the teenager when listening to the parent or the teacher drone on about something.
the image, if you can't see it, it does look a lot like when my 5-year-old tries to draw the letter M and just never knows when to stop going up.
Crisscross, crisscross. Is it M's? Is it X's? Oh, look, there's lines crossing that way. Yeah, this is...
That definitely looks very deliberate, though.
Absolutely deliberate.
Yeah.
And actually, if you're just engravings, there's, I think, something that even dates back to like 500,000 years that they have found at a different location where they could find what looked like intentional zigzag marks carved into a shell.
It is no buffalo, but it is, it's important, and it's 73,000 years old. So this is
cool.
Pretty cool.
Taking it back.
And it was discovered by researchers at Witwatersrand. Am I saying this right? I always mess this up. Which is the same people who are the same university out of which the study is going on into Homo naledi.
Oh, fascinating. Very cool. Doing some good work there. Oh, that was some good work. We're moving into the next segment of the show where there's lots of good work coming out of the animal kingdom. Blazes Animal Corner.
With Blaze.
Doves are creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Yeah, it's Blair.
I missed it. I should have. Kept the theme.
All right. Well, let's start with a case of kidnapping.
Oh, geez. Okay, I like a good story.
Yes.
What's up?
This is a
This
case
segment
of
always, like,
this is the case of invertebrate kidnapping. If you've ever heard of pteropods, starts with a P, pteropods, right? They are what the aquarium I used to work at called the potato chips of the sea. If you're a salmon or many other animals in the ocean, you can't have just one. You're popping little pteropods all day. They're the base of a lot of different food chains in the ocean, and they're extremely important. And the reason I've heard of them before is because they are affected pretty heavily by ocean acidification. But this story isn't about that. This story is about a case of kidnapping of pteropods. So in this study, there were some videos captured of other invertebrates, of amphipods, which are like little shrimpies, grabbing pteropods as they moved through the ocean. So pteropods, they, aside from apparently being delicious, they have to do something about that or they would be eaten all the time. They actually produce chemical deterrents to ward off predators. And so it looks like amphipods are actually grabbing these guys to kind of spray the chemical deterrents and use that benefit for themselves.
Huh.
The reason we think this is probably not a symbiotic relationship and probably a case of kidnapping is that the pteropods are grabbed in such a way that they can't move at all. They can't move their wings. They can't move their feet. They are unable to feed. Yes.
They're trapped.
They're trapped. So amphipods are also popular food items. Think about how much people love to eat shrimp. They're very popular prey for fish and seabirds. This was a discovery where by just taking pictures and video, they saw amphipods carrying something unusual on their backs. And upon closer inspection, they realized it was pteropods. They have U.S. researchers actually had already found this back in 1990, but only for high Antarctic coastal waters and not for the open ocean, which is what they found in this. And they found enough cases that they think this is a pretty normal occurrence. They found it in 2 different species of amphipods, and they were carrying different species of pteropods. Their sample size is too small to say for sure whether it's a species-specific pairing, but that's what they found. But it was only a few cases, so it's really hard to say for sure that they're that specific. So cod, ice fishes, and other predators were in fact deterred by chemicals that the pteropods were producing as they were being carried. And when amphipods take those pteropods hostage, the amphipods are not affected by the poison, but these other animals, ice fish and others, they avoid them because they don't taste good. So their original thought was that perhaps the pteropod benefits in some way from saving energy by being carried. Maybe they don't have to swim, and so it's not so bad. But they actually found, since the amphipod uses both, actually two, not all, but two pairs of their legs to keep the gastropods on their back, they're completely unable to move around, hunt, gather food, anything. So they call that kidnapping. The reason this is so interesting, besides just a really unusual kind of species pairing that I've never really heard of before, is, as one of the lead authors states, quote, "We're probably overlooking numerous such associations between species because they are no longer visible after net sampling," which makes sense. If you're sampling from the Antarctic and you're just using some sort of net or trawl, when you pull something up, a lot of things might fall apart or not be displayed as you anticipated. Quote continues, "In the future, we will hopefully be able to use suitable underwater technologies with high definition cameras to investigate even the smallest life forms in their habitat. This will provide insights into the numerous exciting mysteries of interspecific interactions, which have so far remained hidden for biologists, but which undoubtedly play an important role in predator-prey relationships in the ocean." So these are two little itty-bitty invertebrates that are being used or using one another in a way that could affect dynamics, ecosystems, food webs in a way we had no idea.
Yeah, that's amazing.
Yeah.
I'm looking at some of these pictures of these animals, like you said, the amphipods are like shrimp, and so they've got the hard carapace, they've got the exoskeleton, but the pteropods, many of them, especially like one of the species in the study, the Cleone lemacina antarctica, it's a sea butterfly, and these look like little angels in the water. They've got a little round head, what looks like a head part, and then what appear to be translucent wings, and so they use these wing type appendages to propel themselves through the water, but they don't have that protection otherwise.
Mhm.
They move
Yeah.
through the water column differently from the amphipods, and so I wonder what else is going on there.
Absolutely. And it only makes you wonder, especially in places that we haven't explored very much under the water. I talk all the time about how, yes, space, lots of cool things to discover, so much still to discover underwater on our own planet, that especially in the Antarctic, especially in the deep sea, there are places that we need to explore with more than just nets, because there are interactions going on that we can't capture by pulling things up to where we are. So that's very interesting.
Yeah.
Another invertebrate relationship that's quite unusual involves digger bees from Habropoda with beetles that trick them, these digger bees, into taking care of their larvae. So this is a study out of UC Davis. What?
Nice.
Hey, Davis, listening on the radio. So I got excited. So this study is looking at the larva of Mello franciscanus beetles. The larva lure male digger bees with chemical signals mimicking female sex pheromones. Little larvae pretending to be female bees. These beetle larvae, also known as triungulans, attach themselves to the males. Oh no! Transfer to female bees during copulation. Double oh no! And then hitch a ride back to the nest, where, triple oh no, they feed on bee eggs and provisions and emerge as adult beetles the following winter. So they have a pretty sweet deal, but the poor digger bees, not so great. This study looked at how there were male bees of different species of diggers. They found that they were attracted to local parasite larvae more than larvae from distant locales. So their fake female sex pheromones are location specific. They tailor their pheromone mimicking blends to the pheromones of their local hosts and the larval aggregation, that's scientist speak for where the larvae hang out. They adapted their location to a perching height equal to the patrolling height of local male bees. So not only are the pheromones species or location specific, but the place where they hitch a ride is specific to where the bees hang out in that area. So this is a case, speaking of evolution, of rapid evolution via local adaptation. So it's difficult to parse out here whether it's species specific or whether it's location specific, but what we know for sure is that it's definitely, from this study, there is a location correlation. Say that 5 times fast.
That's right. The location correlation.
Location correlation. Yes. So this is very interesting for a bunch of reasons, but of course, at the very basic level, this is a good reminder that species interactions can be very intricate and specific to specific areas. And what I'm thinking about with this is things like climate change. When one species moves because maybe they're herbivores and they're following a plant species that has moved, and then their parasites don't move, that's good for that animal that's not being parasitized anymore, but it could be bad for the ecosystem and the parasite. Yes. So it's this whole thing about species interactions where even a slight geographical shift could mean a huge change in ecosystems.
And when we're talking about insects, I mean, this is huge. We've had stories about flowers on or plants on top of mountains that are changing their distribution as a result of climate change and the melting glaciers and all the things. And bees are having to forage elsewhere, having to find different plants for their foraging, having to figure things out. And so,
Yeah. So in this case, if the digger bees moved elsewhere, these beetles would be in dire straits.
There's an interesting link on the NOAA, NOAA site, which is also a good place to look at hurricane satellite imagery if you're interested in that sort of thing right about now. But they've got an interesting their main web page has a few global warming links. One is a sort of ask a scientist bit, and the other one is a map of showing where in America, in the United States, spring is showing up earlier than it normally does. And it's a wide swath. It's not evenly dispersed. It's a sort of interesting map because it's, of course, related as well to weather patterns. But it is showing this huge swath through the United States where spring seems to be showing up earlier each year. And of course, this is also what can dramatically affect if bird migration is taking place, but the insects aren't there, or the flowers aren't there, or the flowers are already there and they're past the stage where the insect would want to, or the bird would want to. It's that whole
and that's a very good point.
If the pollinators aren't there, then the plants don't get to reproduce, and then the frogs in the log and the toad on the hole and whatever.
So in this case, I was talking about a geographic shift, but there might be a temporal shift that could cause a problem with this parasite and host relationship that we wouldn't anticipate. Maybe these bees adjust to a new flowering cycle, but the beetle does not. Absolutely. That's a good question.
And what was interesting about that too is like we think of incremental change as how we will experience the coming effects of global warming. But when you see how large a territory is being affected by this temperature change, you realize, oh gosh, that's going to happen a lot of places a lot faster than incremental. These could be
the increments are much smaller.
It's still
it's dominoes.
But yes,
it's when you hit those dominoes, it might take 5 minutes to get to the other end. But there's all those other dominoes along the way. That's a lot of
increments. I guess you're right.
Yeah.
Beetle, tree, me. There we go. Dominoes.
Yes. I will end the animal corner just because we're in the second half of the show anyway.
No, don't end the corner.
It's time to do quick.
Yes, my very quick story is how I'm going to finish up the animal corner. It's a bit of hope for your Wednesday evening or whenever you're listening to this. It's about sharks. Sharks, as we know, have a PR problem. People are terrified of them unnecessarily. I could go on and on about how more people are killed every year by cows than sharks, blah, blah, blah. Anyway, this is an interesting study looking at people who have participated in shark ecotourism programs. They surveyed 547 participants, and they wanted to see, this is in Oahu, Hawaii. They looked at participants' knowledge of attitudes towards sharks and their intention to engage with shark conservation projects before and after. They compared those with 488 members of the public who had not been in shark ecotourism. So this is a decent sized study. Before taking part in shark ecotourism, the people who had signed up, people who were going to these shark programs, were generally more environmentally minded, more knowledgeable about sharks, and had more positive attitudes toward sharks. That makes sense. You're probably not going to jump in a shark cage if you're terrified of sharks. 71% of participants had positive attitudes toward sharks before the tour, with only 45% of the general public. So a lot more. However, even though they were more interested in sharks and the sharks were less vilified before the ecotourism program, afterwards, participants had significantly more knowledge of the ecological role of sharks and a more favorable attitudes towards them. There was a 39% increase in knowledge, and 97% of participants who had negative attitudes changed their minds to a positive one. 97%. So ultimately, these programs had a significant positive effect on people's intentions to engage in shark conservation behavior. And so they were not just preaching to the choir or preaching to the converted. It did improve perception and potentially help sharks. But the kind of disclaimer on this is that there are still legitimate concerns about forms of shark tourism and whether it is a good one or a bad one. Ecological impacts, public safety, how they chum, if they chum. So you still want to do your homework and make sure that if you want to promote or go on a shark ecotourism experience, you should make sure it's a good one, that it's accredited or vouched for by other organizations that you trust. But ultimately, this is not a surprise to me, a person who's in environmental education, because generally speaking, the more you know about animals and the more time you spend with them, the more you care about them, the more you care about them, the more you'll do to help them. That's the whole basis of, I don't know, my entire life's work. So like, it's not super surprising, but it's really good to see that even sharks, this animal that's very polarizing, people are getting a positive impact from spending time with them and learning about them.
Although I do still think that you give cows a bad rap.
They're fine. They're just not as dumb as everyone thinks is the thing.
The cows and the squirrels.
Yeah.
Yeah, but true. But the exposure also very often, it's not just the getting people to care about things. It also is reducing fear.
Oh yeah, absolutely.
So education and knowledge reduce the fear of the unknown because it's no longer unknown.
Yeah, I mean, I take snakes into classrooms all the time, and it usually happens with high schoolers that they're terrified of them. They like want to go to the other end of the room. And if I can coach them into finally touching the snake, they're all tense, they're really scared. And you'll kind of see this moment where once they touch the snake, nothing's really changed, but they touched it and they didn't die, I guess. They kind of relax a little bit. Their shoulders go down. They ask questions about the snake afterwards. It's a very clear shift from this moment of mysticism and fear to this interest and curiosity.
And I think that fear is human generated and not something innate about the animal itself. My youngest,
well, except for snakes and spiders, there's research.
We did research on that. Yeah, we talked about the research. Yeah,
my youngest, she would always approach snakes and spiders, not a problem, until her older sister told her that they were scary, and then was scared of them. Right. But before that, no fear.
My last stories for the night. I got a bunch of quick ones I want to run through. Researchers looked at urine from dolphins, and what they found
That's got to
is
be difficult to collect.
right, collecting urine samples from dolphins. This is studied in the Journal of American Geophysical Union. These researchers from the College of Charleston and Chicago Zoological Society determined that there were phthalates in the dolphin urine. What does that mean? Plastic byproducts in the urine of dolphins. Is a surprise? No, not necessarily. Do we know what it means for dolphins? No, we do not. We just know that this is in their system and is the plastics are in their system. We know that phthalates have reproductive effects in humans at certain levels. So the question now is that happening in dolphins as well?
But
Are there
wait,
reproductive issues resulting from the plastic they're swimming in?
Where and when, how were these dolphins collected? These aren't captive dolphins then. These aren't like
No, these are wild dolphins.
Aquarium or anything. No. Okay.
No, these were wild dolphins, and so this is wild dolphin urine.
That is
Wild dolphin urine.
Wild dolphin urine. Yeah, we don't know the health effects. We don't know what it means for their reproductive systems. We don't know what it means for the downstream population effects.
Why
we don't know
don't we
anything.
know these things?
Because
We
this
should
is
know this.
because this is the first time anyone has started looking at these questions. So now we know that the phthalates are there. So that's all we know. Now we know to look further. So it's the beginning of a journey into the world of wild dolphin urine. Beyond that, researchers have been looking for ordinary matter in the universe. There's been a question of where's all the matter? What's the matter with the matter? And there was a story I reported on a while back. Some astronomers taking various pictures of bright stars and galaxies in the universe, pairs of galaxies that were close to each other, and kind of layering them on top of each other so that they could image and increase the signal on the hot gases in between the galaxies, enabling them to come up with, hey, we found all of the missing matter. It's not really missing. It's just not in stars. It's not in the galaxies and the planets. It's in between. It's in the hot gas. Well, there was a question as to the accuracy of this system and whether or not there was a bit of noise in there. So other researchers used a different tact in which they looked at in a paper that was just published, they looked at signals from a quasar, a very distant quasar. And this is a spinning star that basically acts like a lighthouse. And the light energy that comes out from this spinning quasar illuminates the space around it. And so they looked at the space that's illuminated by the quasar. Lo and behold, dun, 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 missing matter. And they looked at not a hydrogen signal, which you would expect, but they looked at an oxygen signal to do this because of various chemical interactions that would strip electrons out of the way from hydrogen, making it not signalable. So anyway, oxygen became a tracer that they were able to look at, illuminated in the gas by this quasar, and their numbers corroborate the findings from the previous study that I reported on, missing matter. It's in the space between us, our spaces. It's in the tendrils and the spaces. Yes, it's in between. It's hot gas, it's dust, it's stuff. It's out there. It's not missing anymore. We found it. Another space story, researchers in 2017 with the Breakthrough Listen project reported on 21 repeating light pulses, fast radio bursts. They're like, looking at this particular dwarf galaxy FRB 121102. And these fast radio bursts occurred within 1 hour. It was an amazing amount. And researchers were like, what would be doing that? Why would that happen? And this dwarf galaxy has this repeating signal that happens. It keeps happening. And so they don't understand. They think maybe because, why not? Aliens. And
Or
so
neutron star.
yeah, it could be a neutron star also. This particular star is about 3 billion light years from Earth, so not anywhere close to us. But researchers wanted to take another look at that data and really see what was going on. So they threw artificial intelligence at it. The researchers at University of California, Berkeley SETI Research Center used machine learning and applied it to the data set, and they found even more light flashes. Not one, not two, 72 additional light flashes from whatever this source is. So
Okay, I give up. Now it's aliens.
yeah, 93 fast radio bursts from a single source within the span of an hour on that day. And yeah, nobody knows what they are. Nobody knows what it's from. It's in a galaxy far, far away. And then my final story for the evening.
Wait, that's the end of the story. I'm like on the edge of my chair, wanting to know how this ends, and you're like, and I still don't know what's going on.
No,
that's
but
really the end of it. Oh
that's really they don't know what's going on. But the exciting thing actually is the application of machine learning techniques, applying them to the data set and uncovering these previously undiscovered signals.
Yes. A deeper mystery is always more exciting than a resolved one.
The mystery continues. And finally, my last story. Have you ever been hypnotized watching drops of hot water skitter across the surface of drops of water skitter across the surface of a very hot cast iron pan?
No.
Have you ever dripped water on a pan and just watched those drips of water do a little dance across the surface and wondered, how do they do that? You haven't?
I mean, I've seen it.
I'm totally calling you Blaze from now on.
That's how I used to do my dishes.
I know.
I mean, it's neat.
Water and boil it off. Then the whole pan will be clean. That's how that works.
The pan will be clean. You boil it off. All right. So this effect of the droplets of water skittering across the surface of a hot pan is called the Leidenfrost effect. And this doesn't only happen on the surface of hot pans. It happens other places. But this effect, researchers have been looking at for a while. And they're like, oh, these drops of water, what's going on? And they showed before that what happens is because of the heat and it's water, there's a little bit of steam that builds up under the drops, allowing them to levitate almost. Kind of like a hockey puck over ice, or those stones that slide across that one particular lake and leave.
Oh yeah.
Yes, kind of like that. So anyway, the drops of water, there's a little bit of steam underneath them, but that doesn't explain the dance and the movement and why they're directed in certain ways over the surface of the pan. And so some researchers decided that they needed to, of course, get up close and personal with drops of water on a hot pan, and they injected water drops with little glass beads so they could see what was happening inside of the water drops. And what they found is that there are all sorts of little currents and eddies inside a single drop of water. And those currents and eddies, they flow as the droplet evaporates over the surface of the water. And at first, it's very chaotic, not very ordered. The glass beads that are dropped into the water move conveyed by these different eddies, bouncing from one eddy to another, maybe rotating around the droplet of water. And so they're finding that there's this very dynamic interior to that little drop of water that's skittering over its levitator of steam over that hot pan. But eventually, those chaotic eddies turn into a sustained flow in one particular direction. And that sustained flow is what can drive the droplet of water to like just basically drive across a hot pan.
Wow.
And
This is insane.
it's pretty intense. Let me tell you.
Yeah. So the image that's there of this droplet, it starts out as though it was rendered by Van Gogh's Starry Night.
Right?
And then it turns into Jupiter.
Indeed.
It's really fascinating.
And it's spinning and spinning and spinning. And so potentially looking at this, maybe it could tell us about Jupiter's atmosphere. Maybe it could tell us about
It's got bands.
It's
This looks like look. If you told me, hey, look, we got some footage of a planet that's a gaseous planet with bands on it that's spinning fast, I'd be like, wow, they actually have footage of an exoplanet. How amazing is that? Because that's what that looks like. I would not have imagined that was a drop of water.
But it's a drop of water.
Wow.
That is
And
super
so
cool.
they found this imbalance inside that causes it to move in the same direction. That's like it's like a miniature tidal wave that moves the droplet in the direction of that flow, and it rolls them toward a cooler part of the heated surface. The results suggest that maybe this kind of movement could be used to control a device and lead to new self-propelled devices.
Wait a second. Could this be operating with like a kinetic energy, like a gyroscope?
Maybe.
That's so wild. At that level, that would be insane.
It would be insane. Yes. So there's very exciting stuff going on in those little droplets. Yes. Drops of quicksilver, drops of water, little tiny Jupiter in your pan. That's right. It's very interesting stuff. Super exciting. And that's my last story. Did you have one about
I got one more.
super blood? Yeah. What's going on?
Wolverine, the mutant Marvel comic book character with the superpower of being able to rapidly heal.
Yes.
Which, think about it, that's a really good superpower.
It is.
So he's in the fight with the super bad guy, and then he gets hurt. He gets injured. He grimaces. But in the next panel, he's fine again. The next frame of the comic, all healed up, ready to go do more super mutiny mayhem. Turns out we all have the same ability as Wolverine. We're just a lot slower at it, right? It takes us longer to heal. Like we get cut, we heal up too. That's not really, it's just the time frame which makes it a superpower. So we get cut, wound heals itself. It takes a little time. We get the blood platelets. They go in there and they kind of create a scaffolding, a scab. Skin comes in, skin cells close the wound. Before long, we've got a healed wound, maybe a little scar at the end, a little trophy of the entropic event. But that process might be somewhat stranger than we initially thought. This is out of the research out of the Oslo University Hospital, led by Emma Long and Stig Alvboe. Oh, I said Boa. It's not right. They performed experiments on wound-free skin cells that were put into contact with blood. The result? The skin cells, again, wound-free, no cuts. The skin cells started to move and grow together in the same direction as soon as blood was introduced to them, indicating somehow blood is signaling skin, or skin is looking for blood as the signal to start a healing process separate from everything else. This is pretty wild.
Yeah.
So
I had understood, there's histamines and like you scratch your skin and there's a release of histamines and there's various things inflammation factors in the skin that respond to a wound.
What is inflammation? What is inflammation? It's an increased blood flow, right?
Right. But this is beyond that. This is something that's in the blood itself that we don't know about
in
yet.
the blood, but it's also a reaction in the skin. So they they did a couple little narrowing downs of the experiment. They introduced blood to sort of disparate skin cells, and they found they sort of moved randomly in their reaction. But then they would add blood to a more connected group of skin cells, and they found that they moved in a coordinated way. They sort of moved in unison. They could get these skin cells to span micro and even millimeter length distances in their migrations. So there's something of an awareness within the skin cells. And I say awareness from a totally non-consciousness. But there's a trigger within the skin cells that's looking for blood as it's go time to do the healing mechanism, separate from anything else.
So let me ask you this: does it have to be my own blood?
Okay.
Well,
Right.
there's the immune response if it's not your own blood.
Right. Well, but if you get the right type of blood,
Yeah. What if I got the right type of blood?
Yes, and then filled up a bathtub full of it so that you could rejuvenate all of your skin cells. I don't know where this is going.
I really like the phrase "blood regulated skin dynamics."
Yeah.
I kind of find that an enjoyable phrase.
Yes, I'm going into my blood regulated dynamic submersion. I'll see you in an hour.
Right. Well, you ask about the, bath of blood or whatever. I mean, there's a sensationalized article in the pop culture media today about drinking young people's blood is the tonic for fighting aging.
Oh no.
We've done those studies before, but I mean, this is something.
Well, they're true, but you don't drink it. You get a transfusion. Come on, people, get it right.
Get it
I'll
right.
just make you barf.
But maybe you just need to cut yourself and lie in a bath of blood. No.
Bloodletting. See? We thought they were wrong. Turns out they were right.
This is going dark places. Blood.
So, Professor Bo, one may speculate based on our data that cell migration is also activated in situations of interaction with blood. We may also speculate that our skin cells are much more active and dynamic than previously thought, and that blood-regulated skin dynamics occurs in many different situations. But there's a interesting independence there, I think, where the reaction is very localized to the wound. It's not the brain. It's not a whole complex number of chemicals that need to go seek out. Like one of the pictures that I have is this weird blood cell, which blood cells are very weird. They have, with little legs walking down a vein and then looking, sort of probing around for, and then finding a cut or a break, and then sort of plastering itself in the hole to stop it.
It's fascinating though, the connectivity between the cells and the blood. I mean, whatever's in the blood. I mean, there's energy in the blood. There's oxygen in the blood. Blood removes metabolites to clean up an area, make cells work better. I mean, the blood is making the skin cells want to move, be dynamic,
want to heal, want to stop it from escaping.
Yeah. Makes me want to move it. Make me move it. And we're going to move it right to the end of our show. We have hit all the stories that were in our rundown. Didn't drop a single one. It's been a long show, but oh, so good tonight. So good. So much fun. Everyone, thank you so much for listening. Thank you to those of you in the YouTube chat room, in our This Week in Science chat room over at Freenode. Thank you for being a part of the chat, for conversing during the show about the various things that we have discussed here tonight. Thank you to Fada for helping out with monitoring our YouTube chat room, for doing social media and the show notes over on YouTube. Thank you to Gord McLeod. Thanks for monitoring over in our regular chat room over there. And Identity4, thanks for recording the show. I really do appreciate that. And finally, shout outs to our Patreon sponsors. Thank you to Paul Disney, G. Burton Lattimore, Richard Onimus, Ed Dyer, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Andy Gross, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, John Ratnaswamy, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard, Brian Condren, Aiden Jeff, Ben Bignell, Richard Porter, Jacqueline Boyster, Noodles, Kevin Reardon, Dave Neighbor, Christoph Zucknerek, Noah Zemke, Jim Seabright, Ashish Pants, Ulysses Adkins, Sarah Chavez, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Sluzewski, Jim Drippo, Greg Riley, Sean Lamb, Ben Rothig, Steve Lesseman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, EO, Mark Tyrone Fong, and Keith Corsell. Thank you for all of your support on Patreon. We really do appreciate it. And for those of you who would like more information, you can find information at TWIS.org. Click on that Patreon link, or you can go directly to patreon.com/thisweekinscience. Remember, you can help us out a lot just by heading over to TWIS.org and telling others if they're interested about that website. On next week's show, we're going to be talking about plastics in the ocean. We're going to be talking about remediation of plastics in the ocean. You know, there's a big experiment right now looking at this boom arm net thing that's supposed to grab the plastics. A lot of people aren't happy.
PhD project.
Yeah, a lot of people are not happy with this particular remediation experiment and not looking forward to the potential of what it might mean for the ecological habitats. And so we're going to be talking with a woman, Kathryn Matthews from Oceana, this next week. And I'm very much looking forward to talking with her about using scientific evidence to manage our ocean habitats better, and talk to her about the organization she works for and find out a lot more. All right, everyone. Next week, Wednesday evening, 8 p.m. Pacific time, we're going to talk about the oceans. Be there if you can, twist.org/live. But if you can't make it, it's all right, because we are on the internet. You can find past episodes at twist.org or at our YouTube channel. Just look for This Week in Science.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can look for TWIS, the number 4 Droid app in your Android marketplace, or simply This Week in Science and anything Apple marketplacey.
Looking for more information on anything you've heard here today? You can find show notes on our website. That's at www.twist.org. While you're there, you can also make comments and start conversations with the hosts or their listeners.
Yeah. Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in your subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a "What has science done for me lately" song, please let us know.
We'll be back here next week, and I can't wait. We hope you'll join us again for more great science news.
Yes. But if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science. science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma. Got the eye. Cause it's This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 This Week in 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 Science.
And we have come to the end of our show. Don't you know? Don't you know? Yeah. Oh, you want the sugars in space research, old scratch? I'll get you a link. Just you wait a second. I'll stop screen sharing. Just you wait a moment and I will get you that research because I have links. I have links in my rundown. Links in my rundown.
What are you looking for?
An article about the space sugars for old scratch in the YouTube chat. So here is the, let's see, I put the link. How to put a link. There's a PopSci explanation link. And then the paper. Here is the paper. Eckhart et al. Gas phase sugar formation using hydroxymethylene as a reactive formaldehyde isomer. Dun, dun, dun. Gas phase. Gas phase.
Okay, so I have a proposal.
Okay, first, 2019 Blair's Animal Corner calendar is being colored and is coming soon. She has a cover.
I have a cover, which means we can take pre-orders.
We can take pre-orders. We can make this happen. Yes. Send me a picture. I'll make an order form. People can make pre-orders.
Yeah.
And then when Blair has finished the calendar, we can mail them to you. And
So
yes,
I am like very close to being done with the line drawings, but I still have quite a bit of coloring to do. But it'll get done. I'm not concerned.
it'll get done.
Yeah, fun. Just some books on tape.
So it'll be ready by October?
I would say November.
November-ish.
As long as it's before the end of the year.
No. It'll be. My hope is we make the mass order at the beginning of November.
Because most people commit to the next calendar before the year ends.
Yes. So that's why the pre-order would be nice. We'd know how many to order.
You know, calendar ordering is like Christmas. It's coming sooner every year.
It's true.
You really got to get in there.
You think so, but then we don't.
I have to make it. It's so much work.
It is a lot of work.
It's not work that I'm doing, so I'm suggesting it should be hurried.
Yeah. Yes, it's all those friggin' dots.
Oh my goodness.
Those dots. So I have a proposal.
And now you have a proposal. Yes. Okay.
My proposal is that we do my unboxing. But I know that you want it to be a separate video, so we would have to tell our web chat to sit tight and terminate this video and start a new one.
Wait, say again?
We could do that.
Okay.
Did it arrive?
It arrived.
Is that a Marshall?
Oh my gosh, and you haven't opened it?
No, I've been waiting like a good girl.
Okay, so I don't know that I necessarily subscribe to this. I think that there should be an outdoor natural.
No, I agree. But also they say that so a couple things. So one thing, the longer you wear them, the more effective they are. Second thing, the idea is I'll explain a little bit of my background with color blindness. I will unbox it, show everyone what's inside, and then everyone will need to stay tuned for an upcoming video of me trying them on outside.
Wait,
and running around and looking at colors.
How did you already make that video if you're just now unboxing?
Yeah,
I get it.
That's what I'm saying. Do an unboxing video right now.
Okay, so I kind of see. I want to be there live. That's the thing. I know it's rough. I want to be there live. It's just too hard.
It's too hard. It's too hard. We can't be there.
I mean, that's the question: is how would I do a hangout? I guess I could do a hangout outside. I guess it's possible.
Do you have your internet?
Yeah, I mean, I guess I could just use cell service.
Could you use your phone as a Wi-Fi?
Yeah, I could do that. So yeah, we could do that on like Sunday or something. But but I would like to do the unboxing separate.
Hot Rod, open it.
Yeah, I know. It's been
there is like I would want to do this next week.
Yeah, Bleak just realized what it is. It is color blindness glasses.
Yes.
Yeah.
Yes, it is.
I would want to do the initial unboxing. This is my desire to see. Live on the show, knowing that it won't be full effect, and then making you this is so cruel. Making you put them away until a Sunday where you can or the next day. I don't have to be there for that. Right, where you go outside and we have the Blair short going out into the world on a sunny day with the glasses on.
Yes.
But I feel like the unboxing should be putting them on and getting an initial indoor reaction. That's how I feel about it.
Ed says we should make the video a short that goes to Patreon contributors first.
Oh my
That's
goodness.
a good one. I also think that you should definitely get accustomed to the glasses before you finish coloring in the calendar.
No, then there'll be like a really clear. It'll be really clear. Like these were before and these were after.
No, actually, I don't know that it would be. I actually don't know that it would be because I think you've done an excellent job thus far. I would be very interested though in the Blair's Animal Corner in Chroma calendar.
So I wonder if my color choices would be more subtle if I could see colors better. Because I
more
feel like
subtly.
I pick the very bright and more different colors.
You'll be surprised.
Because I can tell they're different.
Yeah. I know. I hear you. The contrast element of it is what you're kind of talking about, and I think that's going to be interesting.
So here's the question, Blair. If you do the unboxing tonight and we get to see it, and you know, you put the glasses on for a second.
Yeah.
Oh, there they are in your face, and then we turn off the video or you take the glasses off. Will you really be able to not wear those glasses again until like tomorrow? Or
Yes.
Yes.
Yeah, I feel like Blair would win the marshmallow test.
Oh,
over
yes.
me every time.
I'm all over that
would be like no
test.
contest.
All right.
I'd be stacking up like 4 or 5 marshmallows. I'd be sitting there having eaten the first one and being like, okay, how long does this study really need to go on? Like, she already won. Can I go home now? I want to go.
I'm very good at. Ever since I was a kid, I was very good at what is it called? Delayed gratification.
Yeah.
I was always very good at it.
I still don't know what that means. Still haven't encountered it.
Okay, so is there? Yeah, we can do that tonight. We can do that. I can start
next week. Tonight's way too soon. I'm sorry, Blair. I'm teasing. Yes, I would want to see it right now.
I can start a new hangout and a new live video.
Yeah,
we can.
Why would we have to start anew? I don't understand.
But so we can finish our hangout now, and then we will do. It will be Blair talking about the Enchroma only. So we have a short. So it's Blair talking about her color blindness. We're talking with her about her color blindness, and she does an unboxing.
Yeah,
and then we turn it off, and we're done for the night. So if we don't have anything else to talk about, then I'll turn off. We'll turn off this hangout and start a new one.
Right.
Unless you don't want to be a part of it, Justin.
No. I'm fine with that. Blair's gonna hate me for saying this, but I would much rather start a show with the unboxing.
We're not gonna do that.
Okay. All right then. Shut her down. Start it up. Let's get. Is anybody? As minions, are you with us? How many? I'm just gonna check. I'm gonna go. Minions in the chat room. I know, Fada. It's been a long night. How many of you actually want to see the unboxing right now?
All of them.
I don't see any. No, I got no responses, and I waited no time for them to collect.
You did it. That's right. Fada's like, are we doing it tonight? Someone named Ashley 3891000 says, open the box. Gord says yes. Identity says yes.
Oh yeah, we got Identity. Gord is here to see this.
Oh,
yes, of course.
a bunch.
Leek
A bunch.
says yes.
Ed.
Noodles. Everybody.
Okay.
Ed is the only one. Ed's the only one.
Ed can wait. Ed's right there with you on the delayed gratification.
God, I assumed it said I can't wait, but it says I can wait.
I can wait.
He's with you. I actually can't because.
But this is just the beginning, and there will be another video short that will be we'll do the video short, and we'll give that to Patreon first, and then we'll also we'll do an interview with the Enchroma people and show some of the video of Blair during a TWIS episode.
There's an episode of Twilight Zone.
We're gonna do
the Eye
this.
of the Beholder.
Yeah.
Where they start to unwrap. It's like, just be prepared for it may not be what you expect.
All right, everybody. So
yeah.
So is there any other news? We have another interview next week.
Give me 1 minute. I'll be right back.
You can hang up. You don't need to just hang up. Why do you need to? Because I'm just going to invite you to a new hangout.
Okay, gotcha. Okay, now I
I'll just
understand.
invite you to a new hangout.
I kept thinking we were going to continue with this one.
No,
Does everybody else have to rejoin a thing? The chat room? Are they ready for this?
Yeah, we'll share a new link. It'll be fine.
Chat room has to refresh. It's not that hard. They know what to do. I think they can figure it out. I just want to make sure we have no other business. Calendar's on it's going to be coming. You have the cover. I can make the pre-order link. And that's pretty much it. We got a show next week. All right, everybody. Yeah. Thank you for joining us for This Week in Science. We're going to have an unboxing video. So we're hanging up now, and come look for us in the next live video that'll be starting in about a minute. Okay, stopping broadcast.